You've probably heard the term DNA sequencing and references to genotyping DNA. Now, let's dive into the defining features of each type of technology. We'll start with sequencing. Most cells in the human body carry a copy of the genome, a set of operating instructions encoded in DNA. These copies of genomic DNA all derive from the single set of chromosomes formed during conception. When researchers take a close look at this library of genetic information, they see a code comprised of combinations of 4 basic units represented by the letters A, T, C, and G. The total set of genetic information for one person is over 3 billion units long. It contains vast amounts of information detailing how and when each type of cell or tissue in the body should behave. Sequencing refers to the process of analyzing precisely which letter occurs at each position along the code. This meticulous process yields very detailed results. It is possible to sequence a single gene, a panel of genes, or the whole genome, depending on the clinical indication. In a medical setting, interpreting this information can be complicated, as most variation in the human genome has little to no effect on health. Still, other variants have unknown significance. However, when a patient's disease is unknown or difficult to diagnose, sequencing a set of genes can provide the level of detailed data needed to solve the mystery. On the other hand, genotyping refers to a streamlined method to analyze a patient's DNA for variants with confirmed clinical significance. Using a technique called chip hybridization, scientists can simultaneously analyze hundreds of thousands of variants at locations in the genome that are known or suspected to correlate with disease. It's more like a matching test to see if the patient's genomic sequence at a given location matches with any of the known sequences correlated with a health impact. This high-level examination of a patient's DNA is calibrated to yield clear answers for a pre-specified subset of medical conditions. An estimated 99.9% of the genome does not vary among the human population. Ordering a genotyping test will hone in on the variable areas of the genome found to encode many health-related traits. Which parts of the genomic DNA sequence are analyzed in a genotyping run? For different conditions, lab experts have selected a collection of sequence variants and locations to analyze based on their correlation with disease, spacing throughout the genome, and unique sequence of letters. Analyzing only regions with those characteristics reduces the time and cost associated with large-scale DNA sequencing. Some diagnostic dilemmas require the detailed approach of DNA sequencing. However, the information yielded from a genotyping test will also result in a map of genomic locations where inherited sequences influence health.